We're going to continue today with cellular membranes and membrane transport. And so let's get into this. So before we talk about membranes, I want to just talk a little bit about lab reports. As I mentioned, I'm going to try to cover a little bit about lab reports every day for the next couple of weeks. So hopefully everyone has now submitted the first short report. I know there were a couple of glitches there, and I'm going to be very lenient around late penalties for this one. So if anyone still has it outstanding and gets it in over the next day, I won't count any late marks for that. So Lab 3 is a formal report, which means it's going to be worth a lot more marks, somewhere around 12%, 14% of your final grade. So this is a major component of the course. This is the report marking guide that's found at the end of Lab 3. And you can see that it contains a whole bunch of different sections. And you're going to get grades for all these sections. So title, introduction, methods, results, discussion, literature cited, and appendix. So what I'm going to do is kind of basically spend a little bit of time at the beginning of every lecture for the next 2 weeks talking about one of these sections to give you an idea of what the report is going to look like. If you've never done a formal lab report before, this is a bit of a beast. It's going to take you a bit of time. So please do not try to complete it all over one night. You want to work a little bit on each of it and then spend some time kind of refining it before you actually submit it. So this gives you an idea of how big this thing is going to be. Most people are going to be about maybe 6 pages written, so that's double spaced. May not seem like a lot, but you're going to have introduction, results, discussion. Some people maybe a little more, some people maybe a little less. And up to maybe another 6 pages that's going to include graphs, rough work, appendix, calculations, those kind of things. So I'm going to, like I said, talk about a lot of these things over the next couple of weeks. What I did want to do today is talk a little bit about the graphing, because this is probably one of the first things that you're going to be doing to analyze the data for this report. And so if you look at that video that I sent you yesterday, at the end of the video, there's a bit of a graphing tutorial that will probably give some of the same tips that I'm giving here today. It tells you a little bit about how to make the graph in Microsoft Excel. You do not have to make your graph in Microsoft Excel. You can do it on graph paper or use another software if you like. But there's a few things I want to point out to you while we're making our graph. So you can see there's a sample graph right there. And a few things to point out. So first thing to point out is that notice I have clearly defined data points. Okay, so notice I'm using a nice symbol here, right? I'm not using a little speck or something like that, but a nice big symbol. It could be a triangle, could be a circle, and so on. And so that's one thing I want to see. Another thing that I want to see is your axes labeled. So notice, for example, this one has temperature, and of course you have the units. So if you don't put the units, I don't know what you're talking about. Is it Fahrenheit, Celsius, Kelvin, or otherwise? Notice the concentration is over here. That's micromoles per liter. That's the same as micromoles per liter. Don't use a little m, use a big M, because a little m is meters, and that's a different unit. At the bottom of your graph, you're going to have a title, and the title is going to be called figure something. So you're going to number it. It could be figure 1, 2, 3, or if this is lab 3, so you might want to call it figure 3-1, 3-2, and so on. A, B, and C are also acceptable. And then you're going to give a descriptive title. And I'll talk a little bit more about titles next week in terms of what they're going to look like. But your title should include what was done. Okay, so don't tell me that your title is, let's say, concentration versus temperature. That's not a title. That's telling me what the axes are. Tell me a little bit about the experiment. So, you know, the what of the experiment. What were you testing? And the other thing your title should always include is the binomial name of the organism that was studied. So we were using beetroots. We want to look up what the binomial name of beetroot was, and you're going to include that in your title somewhere. So that's all for tips today. And like I said, I'll just stay tuned at the beginning of every lecture. and I'm going to be throwing in a few more tips here and there as we go along to help you write your report. So back to membranes. This is where we left off last day. We were talking about the fluid mosaic model. And so the fluid mosaic model is this whole idea that takes the phospholipids and the proteins and puts them together. So I want to talk today a little bit about that fluid and mosaic stuff, right? The mosaic, of course, is the proteins being embedded. I want to talk to you a little bit today about the phospholipids and how they are actually fluid. So one thing I just wanted to mention is how have we had confirmation of this kind of model. One is a special type of microscopy. So this is using a scanning electron microscopy. So remember, that's SEM. And in this case, what they've done, this is a special technique called a freeze fracture. It's really kind of cool. What they do is they freeze the cells using liquid nitrogen or something like that, and then they kind of peel them apart. So you can see there's a knife here. I don't really know exactly how the knife works. I think it just notches it, and then they peel it apart. So picture if you peel something like a banana, the banana peel, of course, is going to come off, and it's not ripping the fruit in half. It's actually coming off at a place where there's a natural kind of area of least resistance. So it turns out that the area of least resistance in a membrane is actually right at the middle of the phospholipid bilayer, between the two phospholipid layers. And you can see, and then what they do is they take it and look at it under an electron microscope, and you can see all these little embedded proteins. So there's the mosaic part. So pretty cool. What about the fluid part? There's another pretty cool experiment that was done. The colors here are not the colors that were used. The colors are actually red and green, but I think the textbook changed them because of people who are red-green colorblind. What they did is they took two cell types. It doesn't really matter which two cell types, but in the experiment, they used a mouse cell and a human cell. And what they did is they fluorescently labeled the proteins on the surface of the cell. And then what they did is put it in a petri dish, add a little bit of detergent, and that detergent allows the two membranes to fuse. And here's what they observed, is that initially they fused, and over about an hour, those membrane proteins were actually able to move around. And they found that they were mixed and mingled amongst one another. So this shows that the proteins can move, and that the membrane itself is kind of fluid and dynamic. Here's a little animation I found on the internet. It's not really amazing, but somebody made it. You can see the little membrane proteins moving around over during the fusion event. So let's talk a little bit more about this fluid stuff. So these phospholipids, it turns out that they can move laterally very quick. So by laterally, I mean they can kind of move back and forth. So you can picture them kind of floating through the membrane left and right and back and forth. They can also rotate, so kind of spin around in place. The thing that they don't really do right here is the flip-flopping. So they can't really move between the bilayer. And that's because, of course, we have these polar heads. And the polar heads are not going to want to migrate through the hydrophobic part of the bilayer. So this is a little animation. I'll play that for you, and you can see the phospholipids. Here they go. They can move back and forth, and you can see the integral membrane proteins doing the same thing. And there it goes again. So cute little animation. Always grateful when people do cute little animations on the internet for me. So a couple other things about these, the fluidity and the membrane lipids. So one question is, how can we make a membrane, let's say, more fluid or less fluid? So one thing we can do is look at the length of the fatty acid chains. So I guess I don't have a note on there, but here's the note. So longer chains are less fluid. So you're probably thinking, what is that all about? That is all about those London dispersion forces. When you have larger molecules, they become more solid or less fluid. Another variable is the saturation of the fatty acid chain. So you may remember we were talking about the saturated fats and the unsaturated fats. And the saturated fats, for thinking about food, we often call those fats. And the unsaturated ones we call oils. So that means these ones here are more fluid. So why is that? Again, it has to do with those London dispersion forces. Over here on the left, the saturated fats, you can see that they're kind of flat, and you can stack them really well, and you can get stronger London dispersion forces. The unsaturated ones, you can't really stack them very well, and so they slip around a little bit more, and they're usually more liquidy as opposed to more solid. So there's a little diagram from the textbook, and you can see they're showing the saturated on the right and the unsaturated on the left, and the unsaturated are a little bit more fluid. So I apologize that the left and right got swapped there with the saturated, unsaturated. I didn't realize that. What else can we do to make membranes more fluid or less fluid? Is we can use cholesterol. So here's cholesterol here. And you can see cholesterol, we've got that 1, 2, 3, 4 ring structure. So cholesterol, therefore, is a sterol or a steroid, and it's a lipid. And it has this little polar alcohol group here. And that little polar alcohol group can actually associate with the polar part of the phospholipids. And so it embeds itself into a membrane. So what does cholesterol do? Cholesterol doesn't necessarily make membranes more fluid or less fluid. It more or less stabilizes the optimal fluidity, so that if you add a little heat, the membrane doesn't melt too quickly. If you cool it down, it doesn't solidify too much. So it's kind of involved in the homeostasis there. I'm not going to get into all the mechanisms on that. It's kind of would make the lecture way too long, but I think there's a decent write-up in the textbook. But just know it's an important aspect of regulating fluidity in membranes, and it's an important part of membrane structure, at least in animals. You find other steroids in other types of organisms that are not animals. All right, so talking a little bit more about things that you might expect on the midterm in a couple of weeks. One thing is one type of question I've had before in the long answer is asking people to describe something around the fluid mosaic model, and so this isn't the exact question. Usually I'm a little bit more specific in terms of what I'm looking for. That might be the beginning of the question. It might say, describe the fluid mosaic model of membrane structure. And then I might say something like, be sure to explain, and then might add a few more things to kind of clarify what I'm looking for in the question. But the fluid mosaic model, what is it all about? So it's all about these two things, the fluid part and the mosaic part. So that means you need to include something about phospholipids and something about the proteins, and of course the structure, so hydrophobic and hydrophilic regions. All these other things that we talked about in membranes are not essential for the fluid mosaic model. So you could talk about glycoproteins or glycolipids or cholesterol peripheral proteins, and they might help your mark in the end, but they're not necessarily fully essential. So I started drawing a picture here. Here's my fluid mosaic model, and you can see what I've done here is I've drawn a bunch of phospholipids. I'm identifying that there's a polar component and a non-polar component to the phospholipids and how they arrange themselves in a bilayer. So when you answer questions in tests, use the language of the course, right? So notice all these words here, these buzzwords: hydrophobic, hydrophilic. You could use the word bilayer, phospholipid bilayer. All of those things are going to communicate to me that you do know what you're talking about if they're used appropriately. Of course, there's the cholesterol and the proteins. The cholesterol is not essential for the fluid mosaic model, but it's just there for completeness. And so, maybe mentioning that the integral membrane proteins are going to have hydrophobic amino acids that help them embed themselves into the membrane. Here's some of the other things there. We've got our peripheral proteins, we've got our glycoprotein, and a glycolipid. And so your illustration skills don't have to be amazing. I was able to do this in PowerPoint. Obviously, you're not going to be using PowerPoint for your midterm, and this took me a little while, but you can draw something and label it and describe it. So make sure you do describe as well. So we'll be talking a lot more about midterms in about 2 weeks' time as we get closer to the actual date. But like I said, I want to kind of drop some hints and ideas to get you thinking about it over the next couple of lectures. So where we wanted to start today was we wanted to talk about membrane transport. This is a really huge part of what membranes do, is that they allow things in and out of cells. So membranes, there's my membrane there. It's got the hydrophilic component or the polar area, and it's got this hydrophobic barrier. So that hydrophobic barrier is going to be particular in what is going to be allowed through. So what is going to be allowed through that hydrophobic barrier? Basically, hydrophobic molecules and small things, right? So, you know, this would include our gases. These are important gases as a part of our physiology. And water is a very small molecule. It's very polar, but water is very, very tiny. And it is found like it's very abundant. So there's always water sneaking through, even though it is polar. It's just very, very abundant. So it can get through relatively easily. Not as fast as polar molecules, but water does get through. So that means what does not pass easily, of course, is everything else. Large things and polar and charged molecules. And of course, they're not going to pass through a membrane very easily. So what we want to do today is talk a little bit about getting these molecules in and out, because of course a cell wants to eat and it wants to drink and it wants to do all sorts of things, and it's important to be able to transport molecules in and out of the cell. So first type of transport to talk about is called passive transport. So what is passive transport? Passive transport is diffusion. So what does diffusion mean? It means that molecules are basically, they move around somewhat randomly just due to ambient heat in the environment. And as they move around randomly, they kind of distribute somewhat evenly. So what does this mean? It means that if you have a high concentration on one side of a membrane, eventually they're going to migrate and become evenly distributed. So you go from a high concentration to a low concentration. So what kind of things are diffusing in and out of a cell? Some of these things that we just talked about in the previous slide. So these two gases, oxygen and carbon dioxide, and some hydrophobic molecules. So diffusion is a pretty basic type of transportation. A lot of things can't really get through this way because a lot of things don't necessarily fit that particular category, being really small or hydrophobic. So another type of transport is, of course, osmosis. So what is osmosis? Osmosis is really just a special type of diffusion. It's the diffusion of water. So we talk about osmosis a lot because, of course, water is hugely important in biological cells. And so I have a little video for you. I'm going to play this for you in a second on the right. And you can see what we have is this little red thing here is basically representing the membrane. So I'll play this for you, and then you're going to see someone's going to add some salt. So you can see initially, we have, this is not playing, there we go. Initially, the water molecules are kind of even, just distributing back and forth. And so here we go, we're gonna add that salt. And now the salt is added. So notice what happened. Over on the left, we have 100% water, right? This is 100%. And over on the right, we've added the salt. So it doesn't really matter what percent it is, it's just less than 100. So the water moves from the high concentration to the low concentration. And actually what happens is the water on this side starts to go up. Eventually it's going to stop because of course the force of gravity is going to slow it down. But this is what organisms do. Now, unfortunately, like we had our experiment 2 kind of canceled, and there was a whole experiment around osmosis. And I'll show you a video or 2 here today that kind of simulates what we were doing in lab 2, which is really unfortunate. It's kind of a cool experiment. So what else can we say about osmosis? It turns out osmosis is kind of an important thing, and how important it is and what situation you're in depends on the type of cell that you're in. So here's an animal cell, and there's actually 3 different scenarios here. So notice in the middle, we have something called isotonic. So what does isotonic mean? Tonic means something like dissolved solutes, how salty something is. And iso means the same. So if you take a look at that little red blood cell in the middle, you have water is going in and water is going out. It's going at the same rate. So that little guy there he's happy. And this is how animal cells usually like to be. They like to be in isotonic solution. That's why we have a body, and the body regulates our salt and all those kind of things so that our cells are happy. There are 2 other scenarios, though, where the animal cell may not be happy. So if you take a look on the right, hypertonic. So hyper, if somebody is hyperactive, they have too much energy. So hypertonic means there's too much salt in the solution. So you can see what's happening is that all of the water is basically following the direction of the salt, and the cell shrivels up and dies. So bad for the cell. What about the other scenario? Hypotonic. So hypo, if you think of somebody who's hypothermic, it means they don't have enough heat. So hypotonic means there's not enough salt. So in this case, there's more salt in the animal cell, more solvents, and the water is going in. And then the cell is kind of, it's getting bloated, and it's probably going to burst. I always remember hypotonic because I think of like hippos, right? The hippopotamus animals, they're kind of round and bulbous. And so I think of the cells, becoming like hippos, and hypo is kind of close to that. But if you're looking for a way to remember which is hypo and which is hyper. Here's the picture from the textbook. You can see they're also using red blood cells, hypotonic, isotonic, and hypertonic. So question is, what if you're a plant cell? And this relates to the experiment 2 that we missed. Plant cells, take a look in the middle. In an isotonic solution, sometimes they become kind of flaccid. So flaccid means they're sort of floppy. They're not dead. They're okay in an isotonic solution, but they're also maybe not as happy as they like to be. In a hypertonic solution, they're plasmalized, and plasmalized is different from shriveled in that you can see the cell membrane actually it gets ripped away from the cell wall. So you probably know this, that if you get flowers from somebody who loves you, what do you do with them? You stick them in water. You don't stick them in salt. Salt is going to kill your flowers. So you stick them in water, and the cells take in the water, just like the animal cell. The cell takes in water, but plant cells have cell walls, so the cells don't burst. They become turgid, which sort of means harder. So that's something that a lot of people do when their vegetables sometimes get a little flaccid. Carrots or celery or something like that. You put it in a bowl of water and you let it soak up the water, and they become nice and crisp and crunchy. So this is where plant cells want to be, and hopefully everyone knows that. You water your plants, you put water in your flowers, and they're much more happy. So I'll show you a video that in a moment here that kind of shows what we're doing in the lab. But I kind of want to talk about a couple of other scenarios here. So here's a scenario where we have an animal-like cell, and this animal-like cell does not have a cell wall, but it lives in ponds. So this here is a paramecium. A paramecium is a protist, and we're going to be looking at them in lab 5, and they don't have cell walls. So what do? These things have something special called a contractile vacuole, and the contractile vacuole actually pumps water out. So watch that video there. It's playing, and you can see where the arrow is. If you watch there carefully, you're eventually going to see, just waiting for where the arrow is, you're going to see sort of this circle, and it's going to contract. There it goes. Ready? There it goes. Boom. And that's the contractile vacuole, basically pumping out. So that's one thing we can do. If we, what's this saying? PowerPoint is contacting the server for information. Hopefully I didn't do something wrong with this. My PowerPoint is frozen. So I'm just going to restart the PowerPoint. Sorry about that. Let's see here. I'll have to reshare that in a moment. PowerPoint crashed on me. That's always fun. Okay, I got to find the document. Okay, so back to maybe I'll skip that Paramecium slide in case the video was messing me up. Okay, sorry about that. Let's go back to, sorry, I got to share that. So let's go back to plants. And this is actually what we were supposed to do in lab 2. So this plant that you're seeing here is something called Elodea. Elodea is an aquatic plant, and they're very nice because the leaves are thin, and often when you put them on a microscope slide, you can see one layer of cells. So if you take a look at this here, here's the cells right here. So here's a cell right there. That's one cell. And there's a little cartoon at the top. And so what you can see here is right in the middle is basically, so this is a vacuole. It's a central vacuole in the plant cell. So it's full of water, and all the chloroplasts are kind of pushed against the edge. And what we were going to do in the lab is we were going to stick those things into some salt. And when you stick them in salt, the water gets drained out and all the chloroplasts start to clump around the middle, and you end up with these clump cells. So that was kind of one of the exercises we were going to do in lab 2. So unfortunately, we didn't get to do that. I'll show you this one here. This is actually a video. And if you watch carefully, this thing on the left is playing, and you're going to watch them plasmolyze right in front of your eyes. There they go. Watch carefully. You can see the chloroplasts are clumping. The water is getting drained out. All right, so moving on. Let's talk about some other types of transport. So we're talking about diffusion, and things get across relatively easily. If you're water diffusing, you have a special name, it's called osmosis. But what about things that can't get across very easily? So you have all these polar molecules, and a lot of food for cells is polar molecules. Something like glucose is polar. So how does that get across? In this case, it uses something called facilitated diffusion. So what's a facilitator? A facilitator is a helper. And in this case, it's a helper protein. So you can see in that little animation, you can see those little molecules are getting across and through the help of the protein. There's a couple of different types of facilitated transport type proteins. Some of them are like channels, and literally they have a little channel or a tunnel that goes right through the middle of the protein. So if this here, for example, is a polar molecule, then the inside of the channel is going to be the right size, and it's going to match the characteristics of that molecule. So polar molecule likes a polar channel, for example. I think I have a picture of one here. There's a sucrose channel protein. This one's called a porin. You can see it's got a cute little name, porin, because it's like a pore. And it allows sucrose across. In fact, it exists as a kind of a triplet, a trimer, we call it, of proteins. And that channel is going to be polar, just like sucrose, and it's going to be the right size for sucrose to basically fit through that little pore in there. Some cells want water to transport a lot more quickly. So plant cells like water, and they want to get water throughout their entire system. And so they actually have something to speed up water, to speed up the osmosis. They actually have these channel proteins called aquaporins. You can see there's a diagram of one there, and the water is kind of squeezing through there. You can see the second example is something called a carrier protein. So a carrier protein is a little bit more like a gate in a fence, right? So it kind of hinges back and forth. So here's an example of one that transports lactose across cells. And I'll show you, there's a little animation there, and you can see it opens up, it lets the lactose in, and then it opens up to the other side. So it's a little bit like a revolving door or something like that. So just a bit of a different design, all part of facilitated transport. So passive transport, facilitated transport, both of these don't require any energy investment, meaning that things are already going from a high concentration to a low concentration. Energy is not required for that kind of movement across membranes. You might need a protein to do it that has a gate or a pore in it. But what about things that are not following the concentration gradient going from high to low? So this is called active transport. So here's a scenario here. You can see that outside of the membrane, there's some sort of food, and we want to get it across. And what we want to do is get lots of it across. And so you're going to use some sort of protein system usually. And the difference here is that it's going to require energy. And that energy can take several forms. Often it's ATP. We're going to talk a lot about ATP kind of more after the midterm. I think I've got a slide or two here somewhere, but it's a big energy source for cells. I think, let me just take a peek. Yeah, I do have a slide there. So here's just a little animation showing the active transport. So active, A for active, and requires energy, and A is also for ATP. So hopefully that helps you remember it. So just a primer, what is ATP? So ATP is adenosine, that's the A, triphosphate, ATP. So there it is. So this thing here is adenosine. So that's a nitrogenous base, adenine, and the ribose sugar. And triphosphate, you can see we've got 3 phosphates, tri for 3. And so ATP, different ways to kind of represent it. You can see there's a little cartoon of it. It's a lot of atoms to draw out. When it breaks down and it loses a phosphate, it releases energy. And this is kind of the key thing going on here. So we're going to come back to ATP and talk about it a lot later. Like I said, after the midterm, we're going to talk more about energy. But you can see that ATP is broken down into ADP. The D is for di because it only has 2 phosphates. So notice, by the way, the phosphate is this big yellow thing. It's a phosphorus with a bunch of oxygens around it. Often in the textbooks, we kind of just represent it as a P like that with a circle around it. And when it's not attached to another molecule, often it's represented as P and a little I. And in that case, they've actually circled it, and that means inorganic phosphate, meaning it's not attached to any carbons at all. So if you see that, it means phosphate of some sort, right? So the most famous of the active transport systems is the sodium-potassium pump. You may have heard of it. It's usually in all the textbooks, and I'll show you how it works. It's kind of an interesting case. It pumps 3 sodiums. So you can see 3 sodium atoms are binding on the inside of the cell. And then there comes ATP. You can see ATP is breaking down to ADP. And that little phosphate is actually attaching to the sodium-potassium pump. So what happens when the ATP goes in? The entire thing hinges. So it says, okay, open the gate. And the gate opens. And that allows the sodiums to be released on the outside. Of course, there's the potassium part. So the potassiums bind. They are on the outside already. And 2 potassiums bind. And the phosphate at that time actually disattaches. And that allows the gate to open back to the inside. And then the phosphates are released to the inside of the cell. And the cycle goes on and on. So the sodium-potassium pump is used in your neurons and a lot of other tissues moving sodium-potassium around. Many, many examples of active transporters. Like I said, that's kind of one of the classics. Very well understood system. So we have passive transport, which includes diffusion and osmosis and different types of facilitated diffusion. And we have active transport, and that requires energy. And we're not quite done here. I want to talk about bulk transport. Oh, it's a funny little animation there. What we're looking at here is kind of individual atoms, right, or molecules. But what I want to do is talk about bulk transport. Bulk transport means you're moving things in large masses. And there's a couple of processes I want to talk about there. So let's talk about bulk transport. We have exocytosis. So exo, by the way, means out. Cyte means cell. And osis means process. So this means the process of getting something out of the cell. Endocytosis means in. So if you take a look at what exocytosis is, you have something inside the cell and it's surrounded by a membrane, and that membrane fuses with the plasma membrane and releases whatever it is. So this could be some waste products or something like that. Endocytosis, on the other hand, is basically the exact opposite. So in that case, the plasma membrane makes an invagination or a hole or a pocket or whatever you want to call it, and it surrounds some food or something else and brings in the cell and keeps it inside a little membrane pocket. So let's take a look at these in a bit more detail. This is actually a little video, and you can see in this case here we've got it's a white blood cell. The white blood cell is munching on some bacteria or some viruses or something like that. The bacteria are digested in a lysosome, a digestive compartment, and then the waste products are spit out. So play that for you one more time. You're going to see some endocytosis followed by exocytosis. So start at the beginning again. Here we go. So there's the endocytosis, the destruction of the pathogen, some digestive enzymes, and then here goes some exocytosis. It's going to spit out the waste products. So exocytosis and endocytosis. Not done talking about these yet. There's a little bit more that we can talk about endocytosis because there's actually different types of endocytosis that are worth discussing, and 3 types in fact. So there's that term again, cytosis, the process of something in the cell. And the first one I want to talk about is phagocytosis. So you may remember we were talking about the bacteriophage. So that term phage or phag, phago means eating. So this here is really cellular eating is what we're talking about here. So you can see we're making this invagination here, this little pocket, and bringing in some food and making a food vacuole in there. So notice, just want to point out a term here. We have this term here, pseudopodium, so pseudopodia are these little projections that are made to surround something. So pseudo, by the way, pseudo means fake, and podium means foot. So these are like fake feet. I more think of them as fake arms, but I didn't name them. And there's the food vacuole. So I think I have a little video of this here in a moment. But you can see there's a diagram from the textbook, or sorry, an electron micrograph from the textbook bringing in, it looks like it's an amoeba taking in a bacterium. So here's a video. I'll play this for you. And this is a video of an amoeba trying to eat some paramecium. Apologies, I didn't know the sound was so loud. But here's the pseudopodia, and you can see they're stretching around the paramecium and working very fast and bringing them into food vacuoles. So these amoeba are really incredible. They can bring very large objects in. Paramecium are not, unfortunately, going to make it. So anyway, there's tons of videos on this. You can Google phagocytosis, and there's a whole bunch of these things on YouTube. And of course, I got to share with you Calvin and Hobbes. If you're looking for a great study break cartoon, Calvin and Hobbes is the one. There are so many great cartoons. And let's take a look what Calvin is up to. He says here he's crawling around the floor. He's got a blanket. So he likes to, he's a kid, he likes to pretend to do things. And it says here, the giant amoeba slides along the kitchen floor, extending a cytoplasmic pseudopod. So now you know what a pseudopod is. It's that projection. The protozoan engulfs a package of oatmeal cookies. And well, there's his mom. And she's like, okay, wait a second. And she says, nice try, go put them back. But anyway, maybe you can enjoy that cartoon a little bit more now that you know what a pseudopod is. So if we have cellular eating, we also have cellular drinking. And so this is called pinocytosis. And so this can bring in basically water or solutes. And there's an image there of the process happening there. I also have a video here. So I'll play this for you as well. I got to skip ahead to 1 minute and 24 seconds. Okay, here we go. And there it goes. So it's kind of hard to tell what's going on there, but it's bringing in solutes apparently. Not as familiar with pinocytosis, but it's not food. It's bringing in liquid and solutes anyway. I don't really know why it looks like it's turning itself inside out, but I thought it was kind of a cool video. All right, so one more process to learn, and this is receptor-mediated endocytosis. So you can think of this here as basically picky eating. So normal phagocytosis is just grabbing things and bringing them in. Receptor-mediated endocytosis actually has a receptor. So this little red thing on the surface of the cell is a receptor and is binding a very specific molecule. So in your guts, for example, there are specific receptors for fat molecules, and they bring them in and they digest the fat in a very particular way. And that's a good example of receptor-mediated endocytosis. Here's the image from the textbook. There's a lot of different things going on in terms of the cellular processes. You can see they're calling this a coated pit. I'm not going to talk about that entirely, but it's how this pocket is made. It's a very, very different process in terms of how it's formed. And you can see there's these special pit proteins that are coating the membrane vesicle. I don't have a good video of that one. I just found this little animation, but you can see there's the receptors, and the receptors are bringing in those little purple pinkish molecules and not attaching to the green molecules. Okay, so time check. We have a few minutes left. That's good. I have a video here to show you in a second, but there's the three processes: phagocytosis, pinocytosis, and receptor-mediated endocytosis. So, quick test yourself question. It says, which statements are false? Okay, so let's take a look at these and one at a time. So true or false? Exocytosis and endocytosis are forms of active transport. So we were just talking about these. These are the bulk transport. And so the answer here is, do I have the answer? No, I don't. I got it right on the screen. The answer is true. So bringing in things in bulk actually takes a lot of energy. And I know it's not shown in the diagrams there, but we're talking about not one or two ATPs, but quite a few in order to do that process. B, facilitated transport requires energy. No, so facilitated transport is a type of diffusion that just needs a little help from a transport protein. C, facilitated transport requires channel or carrier proteins. I just said, yes, that's what we need for facilitated transport, so that's true. And then D, osmosis is a form of passive transport, and the answer is true. Osmosis does not require energy. So hopefully you were able to sort that out by reading those. If you have any problems with any of these test yourself questions, please do let me know. So I have a video for you, and this will be where we're going to finish off today. I have these, the textbook has these videos called BioFlex, and they're pretty decent, high quality animations, a little bit of humor sprinkled in there if you listen carefully. And the nice thing is they use the same images that are used in the textbook as well. They're all about 3 minutes long. So I'll play this for you. It's going to recap the stuff we were just talking about on membrane transport. Then I got one or two other things to say, and then I'll let you go for the day. So here goes. Let's play this for you, and hopefully the volume works. Hold on a second here. Make sure I have the volume on. I see some questions. Sorry about that. I totally missed your questions. Somebody's just saying, yay, plants. Yeah, plants are fun. And someone's mentioning about neurons using the ATP sodium potassium pump. Yes. All right, so here we go with the
all our energy and organic molecules from food. Before we can use the molecules we eat, they have to enter our cells, starting with the cells lining the small intestine. Let's zoom in to the surface of a cell. The plasma membrane is selectively permeable. Some molecules can move across it, while others cannot. How do materials enter and leave cells? Lipids, such as these yellow molecules, can dissolve in the lipid bilayer. Notice how they move down their concentration gradient, from where they are more concentrated to where they are less concentrated. This is an example of diffusion. Diffusion is a form of passive transport. It does not require energy from the cell. Most molecules can't cross the lipid bilayer. Here, The sugar fructose moves into intestinal cells by facilitated diffusion, moving down its concentration gradient through a transport protein. Facilitated diffusion doesn't require energy from the cell, so it's also a form of passive transport. Water crosses the plasma membrane by facilitated diffusion, or by diffusing across the lipid bilayer directly. The diffusion of water across a membrane is called osmosis. The sodium-potassium pump moves ions against their concentration gradient, from where they are less concentrated to where they are more concentrated. This requires energy from the cell and is known as active transport. Energy from ATP is used to move sodium ions out of the cell and potassium ions in. Another type of active transport is co-transport. Here, both sodium ions and glucose move into the cell through a co-transporter protein. Sodium ions move down the concentration gradient created by the sodium-potassium pump, and glucose moves against its concentration gradient. Now let's move to the other side of our intestinal cell. Materials can be exported in vesicles that fuse with the plasma membrane and release their contents outside the cell. This process is called exocytosis. In endocytosis, the plasma membrane pinches in, forming a vesicle that contains material from outside the cell. On this side of the cell, we can also see oxygen and carbon dioxide diffusing across the lipid bilayer. Cells use all these processes to get what we need.
Okay, so a couple more things before I let you go. I see somebody's asking, is there a way to access these videos? I've put links for YouTube for these videos in the PowerPoints, and hopefully you can access it there. I don't think they're put up legally, but usually I can find them. If you can't, let me know. I can put the files on Moodle. They're just kind of large files, but usually I can find them on YouTube, and I'm trying to link them into the PowerPoint notes. So take a look in there, and it should be in the PowerPoint. So a couple other things to say about membranes before I let you go. One thing is that the fluid mosaic model has been updated. There's a few updated versions, but they're really just the same old stuff. You still have these fluid lipids, and you've got these mosaic proteins embedded. Mostly it's just tweaks. We're talking about which kind of lipids are on the outside and which kind of lipids are on the inside and whatnot. But other than that, the model hasn't really changed that much. Something else about membranes, we're going to talk about 2 units from now, is that a lot of membranes are part of organelles, particularly if you are in a eukaryotic cell. You've got all these organelles. We've got the nucleus, the endoplasmic reticulum, lysosomes, vesicles, all those sort of things. So we'll come back to that and talk about that in topic 6. As I mentioned, membranes are dynamic. They move things around. And when we talk about the Golgi body and the endoplasmic reticulum, we'll see how things are all transported around the cell. And membranes are important for things like energy production. So if you're familiar with the mitochondria and the electron transport chains and all those kind of things are involved around the mitochondria, and it's essential to have membranes for them. So that's kind of it for today. Next day, we have sort of a mini unit on mostly cell walls and cell surfaces, and we'll also start talking about bacteria.